Welcome to today's lecture of immunoglobulin. Today we will talk about the structure of the immunoglobulin. Before we start to introduce the immunoglobulin, I would like to spend a few minutes to introduce what is the antigen. So antigen, as you know, it is a vast range of substances that can stimulate immune system and can be recognized by the immune response products, including the antibodies, B cells, and T cells. Actually, a native antigen is very large. Not all of the components can bind by our antibodies and lymphocyte. Only a small portion of the antigen can bind by the immune response products. This small portion of the antigen is called epitope or antigenic determinant. As you can see in this figure, in the middle, this is an antigen. It is the entire antigen. And the blue one and white one is represented by the epitopes. Different epitopes, they can be recognized by different type of the antibodies. So this is the specific binding. And a lot of the pathogens, they can work as very good examples of the antigen, such as the virus, bacteria, parasite, fungus, etc. And some toxins released from the pathogens also can work as very good antigens. A native antigen can express several epitopes on their surface. So what is the immunoglobulin? Immunoglobulin has two forms: the secreted immunoglobulin and the membrane-bound immunoglobulin. Immunoglobulin refers to the protein with the immunoglobulin is a protein with some immunological functions. And secreted immunoglobulin, that is the antibody. It exists in the serum and other secretions, such as the milk, tears, saliva, and bile, etc. And the other form of the immunoglobulin is membrane-bound immunoglobulin. That refers to the B cell receptor, which is expressed on the surface of the B cell. So you can see that this structure is similar to the antibody, but it's expressed on the surface of the cell instead of soluble form. And in addition, there was another similar structure, which is called TCR, that is T cell receptor, expressed on the T cells. Although T cells is not an immunoglobulin, but the structure of the TCR is similar to that of the immunoglobulin. Both of them have the antigen binding site, so it belongs to the immunoglobulin superfamily. When the antigen with several epitopes invade into our body, different B cell clones can be activated, and they can produce many kind of the antibodies. So here I want to emphasize that BCR and the antibody generated from the same B cell clone, they can bind with the same epitope. So B cells and their corresponding antibodies, they bind the same epitope. So this is called antigen-specific binding. And antigen with different epitopes, they can stimulate a lot of number of B cell clones. So there are a lot of antibodies produced. Different antibodies, they can bind with different epitopes on the antigens. So that is called diversity of antigen recognition. These two scientists got Nobel Prize for their discovery of chemical structure of the immunoglobulin. They worked independently, and they used different chemical methods to cleave the immunoglobulin to map their biological effect. As you see, immunoglobulin is a very large protein. So if they use some chemical method to cleave the immunoglobulin, some functional fragments will be generated so they can clarify the immunoglobulin functions. So this is the basic structure of the immunoglobulin. Immunoglobulin is a polypeptide. It's composed of the 4 chains, 2 identical light chains shown as yellow, and 2 identical heavy chains shown in red. They form a Y-shaped structure. There are 2 light chains in our body, kappa chain and lambda chain. And there are 5 classes of the heavy chains: mu chain, gamma chain, alpha chain, delta chain, and epsilon chain. If the heavy chain bind to the light chain, they will form the intact immunoglobulin. So mu chain will generate IgM, gamma chain will produce IgG, and alpha chain is for IgA, delta chain for IgD, epsilon chain is for IgE. In our body, there are totally 5 classes immunoglobulins. They are different in the constant region. So what is the variable region and constant region? As you see here, that immunoglobulin is composed of the amino acid. So amino acids folded to form the functional domain. At the amino terminal, the amino acid is about 110 or so amino acid. They folded into a functional domain, which is called variable region, V region. In this region, amino acid show vary greatly among different molecules of the antibodies. And the left part is constant region, because here in the constant region, the amino acid sequence is less variant, and they are called constant region. Variable region contains antigen binding set, while constant region combined with some immune molecules or immune cells. Gamma chain, delta chain, and alpha chain, they contain 3 constant regions: CH1, CH2, and CH3. Between the CH1 and CH2, there is an extended peptide, which is called hinge region. Here is hinge region. But mu chain and epsilon chain, they have no hinge region, but they have an extension CH4 domain. So here is the hinge region. Hinge region rich in the proline residues. Hinge region can give the immunoglobulin more flexibility, so the arm of the antibody can open and close. So they can bind with different epitopes in different locations. They can bind different epitopes in different locations. Okay, now let's look at the hypovariable region. In the variable region of the immunoglobulin, there are 3 portions. They show greatest differences among different antibodies. This is called hypovariable region. And because the hypovariable region structure can match the epitope, so that region is for binding of the epitope of the antigen. You can see that the epitope of the antigen combined with the hypervariable regions, because their structure match each other. And hypovariable region is also called complementarity determining region, CDR. is for antigen binding. And the left part of the variable region on both strands is called framework region. Actually, framework region cannot bind the epitope directly, but they can provide a protein framework to ensure that antigen binding. Treatment with immunoglobulin with different enzymes can generate different fragments, and then we can clarify their functions. For example, if IgG exposed to the papain, and papain can digest the immunoglobulin into 3 fragments, 2 identical FAB fragments. That means antigen binding fragments. So 1 FAB fragment combined with 1 epitope, and another FC fragment combined with the molecules and immune cells to mediate some other infections. If IgG digested by pepsin, a very large fragment will be generated, which is called FAB2 fragment. That means this large fragment, combined with 2 identical epitopes, and the other part of the immunoglobulin will be digested into small pieces without any functions. Finally, let's compare the different classes of the immunoglobulin. So IgG, IgD, and IgE, they exist in the monomer. And IgG is most abundant in the serum. They perform so many functions. They protect us. And IgE is very low level in the serum. Sometimes it's involved in an allergic reaction. So we can see that allergic people can produce a lot of IgE. They are exist in the monomer. And IgA has 2 forms. One, serum IgA is in a monomer. And the IgA in the mucosal system exists in the dimer. IgA secreted IgA is very important for mucosal system, which is composed of 2 IgA monomer linked by J chain and covered by a secretory component. And next, IgM exists in the pentamer in the serum. It is the largest immunoglobulin. It's composed of 5 monomers linked by J chain. And finally, BCR, B cell receptor, on the naive mature B cells also composed of the immunoglobulin. But this immunoglobulin is membrane-bound immunoglobulin. They are composed of the IgM and IgD. But here, IgM is in a monomer instead of pentamer. That's all for today. Thank you for your attention.